Hello all. We've now moved on to chapter 26, the reproductive system. And before we get into the anatomy and physiology of the reproductive system, I want to talk about sexual reproduction itself. Why does it exist? A lot of organisms reproduce asexually, especially very simple ones, single-celled organisms and some multicellular organisms. And this seems to make sense, right? It's easier. Right? There are costs to sexual reproduction. You have to find another individual, and they have to have genes that you think are sufficiently good for you to kind of hook your wagon to. Sometimes just finding a partner is difficult. If you are a mountain lion and you have a solitary range of hundreds of square miles, finding not only another individual, but an individual of the opposite sex that is good enough for your standards, well, that can be difficult. And additionally, if I think I'm very, very good in my environment, right? I am a fit individual. I've had lots of success getting food, right? I probably want my offspring to share all of my DNA, right? Because that means they are likely to be successful too. So why in the world would I want to share my offspring with somebody else, right? To split the DNA and to go through all the trouble that comes with it. Well, the answer seems to be genetic diversity. In dynamic environments where you don't know for sure that the environment in the next generation is going to be the same as the environment in your generation, often it helps to essentially hedge your bets, right? To have offspring with as much diversity as possible. And so then it's likely that at least some of them will be good at exploiting that new environment. And there's a fair amount of research to back this up. There are studies of very closely related organisms, sister species, one of which is sexually reproducing, the other of which is asexually reproducing. And when you compete them in an environment that is changing, that is dynamic, the sexually reproducing organism tends to win out. When you reproduce them or compete them in an environment that is static, meaning it's not changing, the asexually reproducing organism tends to win out. But generally, our environments change. Now you also have, when you shuffle up the DNA, the possibility that you may end up with organisms that are even more fit in a particular environment than you were. Okay, so if we are all sold on the benefits of sexual reproduction, let's talk about how it works. In reproductive systems in humans specifically, you have primary sex organs, which are the gonads. Those make the gametes. So the gonads in men are testes, in females would be ovaries, and they would make the gametes, which in males would be sperm, and in females would be eggs. But along with these primary reproductive structures, the gonads, you have lots of accessory structures. Those accessory structures are glands that secrete things, and tubes that move things along, and some external structures. So again, male gonads are going to be testes, and they are going to produce sperm or spermatozoa for long, and they are going to move the sperm through the male reproductive tract, which is a set of tubes, along with fluid called semen, which is going to help those sperm survive as they go on their journey. Well, what do those accessory organs look like? Well, the ductus deferens is the main tube that carries sperm from the testes over and into the urethra, which will be the final tube. And you have glands, seminal glands and prostate glands, which secrete most of the semen. And then again, the urethra conducts the semen out of the body. And you have the epididymis, which sit on top of the testes and are the site of sperm maturation. There is also some external genitalia in males. The penis, which is going to be what conducts the semen into the female vagina, and the scrotum, which is the sac that the testes hang in, because oddly enough, they live outside the body. And this is a look at that basic anatomy. So let's look at the path of sperm, right? We start in the testes, and we'll look at this more specifically in a bit. But once sperm are produced, they're stored and they mature in this epididymis, right, which is right on top of the testes. And they stay there until it's time for ejaculation. And when it's time for ejaculation, the sperm will move up this ductus deferens, which in the past has been called the vas deferens. That's named after a person. That's why when you get that snipped, it's called a vasectomy. And it comes down over here and down, and the very end of the ductus deferens is this thickened part that's called an ampulla. And it has muscle around it and will squeeze the sperm into this ejaculatory duct, which will go through the prostate. This ejaculatory duct is where it will join with the first bit of semen, which comes from the seminal gland right here. So seminal gland dumps semen through here into this ejaculatory duct. Now you have two of these, one on each side, and both ejaculatory ducts come together into a single urethra, which again is still in the prostate. This is the same urethra that is coming down from the bladder up here and can also carry urine, although not at the same time. We go through the urethra. There are several parts, right? The prostatic urethra goes through the prostate. This part between the prostate and the penis is called the membranous urethra. And then once we get into the penis, it's called either the penile urethra or also the spongy urethra because it goes through the spongy tissue of the penis and then on out. In addition to the seminal gland, which is sometimes called the seminal vesicle here, which produces part of the semen, you will also get part of the semen from the prostate here. And then some of the semen, or really it's pre-ejaculate, comes from what's called the bulbourethral gland. So because this urethra also carries urine, and urine tends to be a little acidic, the urethra can be an acidic environment. So the bulbourethral gland right here, sometimes called the Cowper's gland, secretes a pre-ejaculate fluid that comes out here and kind of clears the way and is a little bit alkaline, and it neutralizes this area here so that you don't kill the sperm. It also lubricates the head of the penis just a bit before sex. When the testes are developing, at first, they are no different from the developing ovaries. It's not till a little bit later in development when differential hormone levels between males and females will cause them to become testes or ovaries. And they start off up in the abdominal pelvic cavity, just like the ovaries would, but they become connected to the base of the pelvic floor by this connective tissue band that's sometimes called the gubernaculum. Gubernaculum, like governor, like sometimes a governor helps direct things, right? This is directing the descent of the testes. And as you grow, for most of the time, this connective tissue band is not shrinking. It stays the same size, but everything else gets bigger, and so the testes end up getting pulled toward the pelvic floor because, this is 2 months. You're pretty tiny at this point. And then eventually it begins to break down and pulls the testes through this inguinal canal into this sac outside the body called the scrotum. Now, this is called the descent of the testes, and the inguinal canal generally closes up more or less, but sometimes it stays open a little bit, and you can get a little bit of the intestines that get sort of squeezed through there, stuck in there, and that is called an inguinal hernia. And while females can get inguinal hernias, because of this inguinal canal that is a little wider for the males for the descent of the testes, little boys are a lot more likely to get inguinal hernias than little girls. So why in the world do scrotums exist? Why are testes outside of the body? They're pretty vulnerable out there, and you could probably ask anybody who's grown up with testes, and they would tell you that it would be nicer if they were inside the body like the ovaries, where they are protected. So why are they out there? Well, turns out the answer is temperature. Sperm are created optimally at a temperature that's just a little bit cooler than the inside of the body. And so they hang out of the body in the scrotum. But you have to be able to regulate that temperature because the temperature outside is not always the same. So when it's hot outside, the scrotum tends to hang further away from the body. When it's cold outside, you have two muscles that tend to pull the scrotum and the testes closer to the body. So the cremaster muscle is around each testes, and it will pull them closer to the body. And then you have the dartos muscle that's in the actual scrotum that will pull the scrotum up. And either way, when it's cold, they're held closer to the body to keep them warm. This temperature differential is why if you're trying to get pregnant and you're having a hard time, they might ask you to wear boxers instead of briefs if you don't normally, because boxers allow the testes to hang further from the body and they can be a little bit cooler. You also have this venous plexus here, or sometimes it's called a pampiniform plexus, of veins around the artery, the testicular artery that comes out here. And that allows you to cool off the blood as it comes out. So the blood coming from inside the body is 98.6 degrees. It's pretty warm. But the blood in the veins has been out here and is on its way back. It's a little cooler than the body. And so when you make the artery come in such close contact with all of this vein, and the vein is spread out into this plexus for more surface area, you have diffusion of temperature. And by the time the blood gets out here in the artery to the testicle, it's actually quite a bit cooler than 98.6 degrees. This helps maintain this cooler temperature, which is optimal for the production of sperm. Now this area here where your ductus deferens and your artery and venous plexus and your nerves are that come out to your testicles, these are all wrapped up in one big cord, one big bundle called the spermatic cord that you can see coming out through here. Right? This is your spermatic cord here. This is this superficial inguinal ring, and this is that inguinal canal right there. So what does a testicle look like if we cut it open and look at a cross-section? Well, most of the testicle is filled with seminiferous tubules. And these tubules are actually where sperm is produced, and we'll talk about that process here shortly. So sperm is sort of consistently produced in these seminiferous tubules. They are surrounded by septa that are made of connective tissue, right? And connective tissue also coats the outside. And you have something called a tunica albuginea, which coats the very outside. That's literally, it means coat of white, right? White coat. So the outside of a testis is white. And when sperm is made in these seminiferous tubules, it then makes its way through the through the rete testis here, through these efferent ductules, and into the epididymis, which we looked at before. And the epididymis is where sperm will mature, and we'll talk about that process again here shortly. And then they will be stored until it's time for ejaculation. The process of sperm production is called spermatogenesis, and it involves three main phases. The first one is mitosis, and you guys may be familiar with mitosis. Mitosis is a process by which you duplicate a cell, really the nucleus of a cell, and then you chop that cell in half, and you produce two genetically identical daughter cells. So you make exact copies genetically of the original cell. So first we have to undergo mitosis. And in mitosis, so we start off, all these cells have chromosomes, which are a line made of DNA, wrapped around little histone proteins. And you have 46 chromosomes in every cell in your body, with the exception of gametes. And you really have 23 types of chromosomes, 1 through 22, and then the 23rd one is the X chromosome. And 1 through 22, especially, all have pairs that are essentially the same. So here, we don't have room to show you all of the, all 23 types of chromosomes, all 46 total. So we're using 6 chromosomes, 3 different types instead. So we'll call it chromosome 1, and we have 2 copies of that. Here we have chromosome 2, 2 copies of that. Here we have chromosome 3, 2 copies of that. Again, in your body, we would have 23 types, each with 2 copies. Well, why do you have 2 copies of each chromosome? Well, simple. It's because you have 2 parents. And one set of those chromosomes, 1 through 23, came from your dad in the sperm. And the other set of those chromosomes, 1 through 23, came from your mom in the egg. And so you have 2 sets. And this is generally what they look like. Before you divide your cell, though, you have to replicate the DNA so that each daughter cell can have a copy of it. So now we've replicated our DNA. We have the same number of chromosomes, 3 pairs, 6 total, but each of them are X-shaped because they have now 2 copies of their DNA. So they have what we call 2 chromatids per chromosome, right? So still 3 types, 6 total chromosomes, but we have 12 chromatids here. Those chromatids are going to get pulled apart during mitosis, and so that the daughter cells have 6 chromosomes, each with 1 chromatid, 3 types each, and they look just like the mother cell.